Hi, everybody. I'm Ann Bryce with Yolo Audubon. Thank you so much for coming tonight. We have a wonderful speaker and a good program, so welcome. I would just like to say a couple of things. One is to remind you that our December is always, we switch dates. So the next meeting, which is the ID workshop for the Christmas bird count, will actually be December 8th. So that's a week or 2 weeks early. Anyway, you'll be getting notice of that, but I just, for those of you who like to get things on your calendar, December 8th. And then the actual Christmas bird count is December 18th, 19th. Ken, which is it? Oops, Ken's on mute. Well, it's on the 18th or 19th. So you can go right now to our website and see who the area leaders are and give them a call and see if you're interested in joining their area and get signed up. It should be hopefully a good Christmas count and not a truncated one like last year.
Right. So the count is December 19th. And in the evening, we will have the compilation via Zoom. That information will go out soon, and with the registration link. And so even if you don't participate in the count, you're welcome to register and participate in the compilation. And if it's a dinner, it's because you brought your own.
Yeah, and the compilation is always fun. It's exciting to see what turned up and how it all went. And the December 8th meeting, the ID workshop, as some of you probably remember all the years, said Steve Hampton, who I see is here with us tonight, did it. Hi, Steve. And it's always our most popular meeting of the year. So please come to that. You'll learn a lot, I promise, even if you're not planning to go on the Christmas bird count. I'm going to turn it over to Ken now to introduce our speaker.
Thank you, Ann. Excuse me. Tonight, we're going to hear about wood ducks, and we have Dr. John Eadie. Dr. Eadie is a professor and the Dennis Raveling Waterfowl Chair in the Department of Wildlife and Fish Conservation Biology at UC Davis. He completed his PhD degree at the University of British Columbia and worked in the zoology department. His next career move saw him joining the faculty at UC Davis in 1995. His research interests include the ecology, conservation, and management of waterfowl and wetlands. His current work focuses on the management and conservation of wetland habitats, breeding waterfowl, mallards and wood ducks, for example, in California, and linking ecological theory to wildlife management and conservation. It uses a combination of experimental observational field studies, molecular genetic techniques in the lab, and population modeling approaches in his research. So please welcome me and please join me in welcoming Dr. Eadie.
Thanks very much, Ken. Hello, everybody. This may take a second or two to actually get this slideshow working because I have to kick it out and then kick it back in order to share it. So bear with me for 1 second, if you don't mind, so that I can share.
Excuse me, John.
Yeah.
I can't remember if Ann mentioned, but folks, we're going to ask you to put your questions in the chat feature at the bottom of the screen. At least it's on the bottom of my screen. If you get your cursor down that way and have it showing, put your chat features in there and we will read those questions off at the end of the talk. Thank you, John.
Yeah, great. Thanks, Ken. Yeah, thanks. Sorry, folks. Give me 1 second here. Bear with me. There we go. That should be it. Is that working, Ken?
It's working.
Okay, good. Well, so thank you everybody for coming to this presentation. It's a pleasure to be here. Just a little bit of background about myself. I hold the Dennis G. Raveling Endowed Waterfowl Chair at UC Davis. So my day job really is, I do a lot of work in population ecology and management, as Ken indicated. A lot of our work focuses on wetland habitat conservation, particularly in the agricultural landscape. I'm very privileged to hold the chair that was named after Dennis Raveling. Dennis was just a giant in the field and died, unfortunately, prematurely of cancer. And so I actually gave up. I just got tenure at the University of Toronto and gave it all up and came down and did it again. So I've learned that you can actually fool a majority of the committee twice. But the Dennis Raveling Chair was set up for to carry on Dennis's legacy, but also to keep a program here in the heart of the Central Flyway, or the Central Valley and the Pacific Flyway. But I have an alter ego. I am a bird nerd of sorts. Particularly, I am a duck nut. And actually, so I guess if we want to put the proper Latin term for that, it would be an anatophile from the family Anatidae. So every good talk begins with a fairy tale. So let me begin mine with this fairy tale. So this is the story of the ugly duckling. And I'm sure many of you have heard this story or read it to your children or were read it as children. And it's a story of a swan egg that is laid in a duck's nest. And of course it hatches out and all the other ducks make fun of it because it looks different. But of course, it grows up and it becomes just a gorgeous swan. I took this story to heart as a child. It gave me great hope that one day perhaps even I could aspire to something. But it wasn't until years later that I ever sort of asked myself the question, how did that swan egg get in the duck's nest in the first place? I don't know if any of you have ever sort of wondered about that as you've read the story or told it to your children. So this actually, it turns out to be a question that has, believe it or not, perplexed me for a good part of my career. And I actually began to sort of think about it seriously as a graduate student back at Queen's University. I was working on these birds, common golden eyes, you'll recognize them. And then as I carried on with these guys, the Barrow's golden eyes in British Columbia. So golden eyes are just fantastic birds. They're cavity nesting waterfowl. So they nest in cavities that are excavated by large primary cavity excavators like pileated woodpeckers. So they need quite large trees and quite large cavities. They also use nest boxes. So that's an important part of the story that'll come in when I start talking about wood ducks. The females, as in most waterfowl, do all the incubating and the young hatch out about 28, 30 days later. And they're entirely precocial. So within 24 hours of hatch, they leave the nest, flying from heights of 20 to 30 feet, hitting the ground and apparently suffering no permanent damage. And the female then cares for them for up to 60 days or so afterwards. So here's me as a young graduate student checking nest boxes in British Columbia with Barrow's golden eyes and common golden eyes. But the story, and going back to the swan egg and the duck nest, began when I was a master's student in Northern Ontario, and we were checking common goldeneye nests. Common goldeneyes normally lay a clutch of, somewhere between sort of 7 to 9 eggs. So here's a typical common goldeneye nest. And then one day while we were checking nests on one of the lakes, we came across a nest of 20 eggs. And I thought, what the heck is going on? I was a complete rookie. I was just starting out my graduate career, and I just couldn't figure out what had happened. And I thought there were some other wildlife researchers that were working with me, technicians. And I remember rowing up the lake to Elk Lake. This is in Northern Ontario, in this canoe on a wavy, choppy day, and thinking, what the heck is going on? I must have made a mistake. There couldn't possibly be 20 eggs. No bird can lay 20 eggs. No duck will lay 20 eggs, and particularly not golden eyes. And I went back to the camp and they said, oh boy, you're in trouble now. Yeah, no, there's something going on here. Yeah, you've made up some numbers or you've miscounted. And I went, oh gosh, what the heck is going on? And then about a week later, we found another nest of 22 eggs. And a week later after that, another nest of 21 eggs. So it wasn't an uncommon phenomena, but it perplexed me. Clearly wasn't the clutch of a single female. Went back to continue my graduate work at Queen's University in Kingston, Ontario, and came across this one paper, this influential paper in 1980 by Yorm Yomtov. It's a paper called Interspecific Nest Parasitism in Birds. Yom Tov laid out this complete idea of this, what until then had been sort of a relatively understudied and almost unknown behavior. And it's a form of parasitism. It's not parasitism like this, where you would have ticks or various kinds of ectoparasites. It's a brood parasite. So most of you, I'm sure, are birders. You're quite familiar with cuckoos and cowbirds. So these are obligate brood parasites. This is a sedge warbler. It's not being eaten by a cuckoo chick. It's feeding a cuckoo chick, and that's the parasitic chick of the cuckoo, European cuckoo, who's laid its eggs in the sedge warbler's nest, and then the sedge warbler provides all the parental care for it. So what a great deal for the cuckoo, and not such a great deal for the sedge warbler. And cuckoos have all sorts of, there's a really interesting arms race that goes on. Cuckoo chicks, when they hatch out, they're blind, they're naked, but they have this instinctive behavior where they'll back up against other eggs in the nest and kick them out so that they can sequester all of the parental care of the host by themselves. So that was relatively well known, that behavior of obligate brood parasitism. What was less well known is this behavior that Yom Tov described, which he called intraspecific, or we now call conspecific brood parasitism. And that's where females in the same species will come in and lay their eggs in the nest of another female and then leave. And so you can imagine there's a considerable benefit to the parasite. She doesn't have to pay for those eggs. She doesn't have to provide any care for them. And there's some cost to the host, who is now providing care for offspring that are not her own. So this was quite a sort of an anomalous behavior. And most of the early papers on this said, well, it's just a mistake. These are females that are confused, or they're laying eggs in the wrong nest. And I just couldn't accept that. I'd already gone through my own sort of epiphany on Elk Lake, roaring through a storm, wondering what was going on. And then Yom Tov's paper really set out a number of ideas about why this behavior might exist, and maybe a lot more common. And it turns out it's found in over 250 species of birds. It's particularly common in waterfowl. But why do females do that? So that led me, and that was something that I worked on a little bit as part of my graduate work. I came to UC Davis, and then more of my work was involved in wetland conservation, wetland management, waterfowl management. But always in the back of my mind, I sort of wanted to return to that question. I felt that we really didn't have a good answer. And that's where the star of our show, the wood duck, comes in. Wood ducks are just incredible. There's been a lot of work on wood ducks going back to the 1920s and before. Frank Belrose, shown here, has published just an amazing compendium of almost everything you could possibly want to know about wood ducks. Gary Hepp, Bobby Kennemer have done some great studies more recently. So you'd think we would know this bird pretty well, but we don't. So this is what we do know. So wood ducks have a very disjunct distribution. Can you see my arrow? Does that show up on your screen? Can somebody give me a yay or nay? Yeah, it does.
Yes,
I
it
see.
does.
Okay, great. Thank you. So wood ducks are most common back east, in the hardwood flooded forest back east. This is a standard. I think this is from the B&A, British Birds of North America publication. So the breeding range is in orange, non-breeding is in dark blue, and then where it overlaps year round is in kind of this purple color. They have a disjunct distribution. There's also a West Coast population with its highest densities, mostly in California, a bit into to Oregon and Washington as well. And they're year-round residents. We've actually done studies. We actually know that genetically these are distinct populations. They're not different subspecies or different. They're not at the level of subspecies, but they certainly have some degree of genetic distinctiveness. So our West Coast population is somewhat unique. What we sometimes forget, I think, is sort of how close the wood duck came to becoming completely extinct. So this is from a paper Frank Belrose's a number of years ago. And he reminds us that in the first decades of the 20th century, it was feared that the wood duck would follow the Labrador duck. Here's Audubon's painting of a Labrador duck into extinction. They were on the verge of extinction. So this is from an article in 1906 from Cook, and he says, so persistently has this duck, the wood duck, they called it the Carolina duck, being pursued that in some sections it has been practically exterminated. And as a result, the wood duck is constantly diminishing in numbers and soon is likely to be known only from books or by tradition. So that was in the turn of the 20th century. A lot of that was due to unregulated harvest. So there were no limits whatsoever. The market hunting birds were shot in huge numbers and sent to market for table fare. There's no regulations whatsoever, and there's also huge forest loss. So these two graphs that I'm going to show you, this is an indication of where the hardwood forests, it's kind of speculation, but where the hardwood forests likely were distributed in the West and the East back in the 1600s. And this is what the estimate looks like in, if it comes up. Whoa, that was interesting. Let's see if we can go back and get that in 1920. So we had lost, huge amounts of that hardwood forest. A lot of it had been, turned into agriculture, and that's the wood ducks require hardwood forest, large trees for their cavities to nest in. That's their habitat. So it's both a consequence of habitat loss as well as overharvest by market hunting. The key aspects in their return was the Migratory Bird Treaty Act of 1918. So that regulated the amount of take, the amount of harvest, interstate transfer of those harvested birds, and started putting regulations in place for the management of migratory birds. And then on the other hand was efforts to try, while we're waiting to grow back the hardwood forests, efforts to try to provide artificial nest sites. And that's where Frank Belrose has played a huge role. Early on, they tried to make them look just like trees, but it wasn't really so successful. And then they found that just regular old plywood boxes would work fine. And there's some debate about how important nest boxes are in the modern breeding biology of wood ducks. But certainly the habitat improvements played a huge role in their recovery. Well, what about our Western population? What about the birds in California? Woodies here have very different habitat. Well, it's not very different, but I mean, the types of habitats are different. Rather than those expansive hardwood flooded forests that you see back east, wood ducks in California are mostly in these riparian regions. And we used to have these vast riparian galleries on every single river. All these river systems that we have in California feeding into the Sacramento and the San Joaquin and all the tributaries would have these vast riparian galleries of oaks, valley oaks, and large old trees. I mean, it's just prime habitat. Lots of shrubs and bush along the shorelines, just ideal habitat for wood ducks. But what's happened is over the years, oops, sorry, I've gone backwards. This seems to be a little bit sluggish on my end. I hope it's coming through for you. We've lost a lot of this riparian. We've just got little narrow riparian strips. Sometimes it's only a tree or two wide. In some cases, there's no trees whatsoever. And even when we do have some riparian with issues of drought and water diversions, this was Puda Creek before the Puda Creek Accord came into play, it can be dry. So we've just had a huge impact on riparian habitats in California. In fact, it's estimated that maybe we've lost 95 to 99% of the riparian habitats that used to be here. So let me just, I'm sure many of you who are bird aficionados, know about the biology of the wood ducks, but let me just share. And I just want to point out, many of the photos that I'm using are my colleague, Bruce Lyon, who's at UC Santa Cruz, and he's a collaborator in a lot of the research that we've been doing. And he's really a fantastic photographer. So woodies, they'll pair up. Actually, they start pairing up in winter, as you probably know. They will be, the female does pretty much all the work. So she'll lay a clutch of somewhere between 10 and 12 eggs. 13 is probably about the maximum. The female does all the incubation. So she'll sit on the eggs for anywhere from 30, sometimes a bit less, sometimes a bit more days. Lines a nest with this lovely sort of a bedding of down. And then the young hatch out after about 30 days, after about a month. And they're just pretty spectacular. So let's see if this video plays. And I don't know, can you hear it? Is this coming through with the sound?
Yes, it is.
Okay, great. Pretty hard, I've a number of students. I teach an ornithology class on campus, and I see some of my students watching. We sort of always are going back and forth about favorite birds, and it's pretty tough not to like a duckling. I mean, they're just incredibly cute. This little gaffer on the top here has decided he wants to be king of the castle. He's determined to get on top of all of his buddies. So that's a pretty fun thing. We get to play with these guys. So they hatch out and then they leave the nest within 24 to 48 hours. So the female goes down, she'll call the ducklings out. She's sort of checking around for predators. And then the ducklings will show up at the entrance and take that grand leap of faith And again, natural cavities can be as much as 24, 30 feet above the ground in some cases. And away they go, leaping to the ground. And then the female gathers them up and she will tend them by herself for, again, sort of up to 2 months period of time until they can fledge. So that's the natural history of the wood ducks. But wood ducks, like golden eyes, have this propensity to do the same sort of behavior where they lay eggs in each other's nests. In fact, they're notorious for it. So this idea of conspecific brood parasitism. So rather than just 1 or 2 females, they'll often get a large number of females laying eggs in nests, and you can get what are often referred to as dump nests. These are nests where you can get 20, 30, 40, 50 eggs in a single nest. So much so that they used to think that females were simply dumping or getting rid of excess eggs. So here's just a shot of one of the nests that we've studied. So yeah, quick guesses on how many eggs there might be here? There's 35. A normal clutch of a wood duck would be 12. And then we find these other bizarre situations. Sometimes we come across 2 females incubating a clutch side by side. In other cases, we come across nests where it appears that the females have literally killed each other. And there's Frank Belrose in his book has documented this as well. So one of the things that we've been doing with one of my graduate students, Catherine Cook, is trying to get a better feeling on sort of what's actually going on in the nest boxes of these wood ducks. And so we've got these little trail cams. I mean, it's just great with modern technology. This is the wood duck's eye, if you were sitting at the bottom of a wood duck nest. And I'm just going to hopefully play you 2 short videos to show you the contrast in how some of these females treat one another when a female comes in to lay an egg in another female's nest. Oops, whoa, having some video. They're not playing very nice. That's those 2 females. Here's another nest, just by contrast. Oops. The fight goes on. Let's move on. So here's a contrast. Here's a female on her nest. Another female comes in. And they're just copacetic. They're just kind of hanging out. There may be a little nibble back and forth. A little bit of a preen. Hi, how are you? So stay with us. Oh, this other female, it's interesting because she's, sticking her head underneath. She's kind of checking out the other female's eggs. What's the state? What are you doing? Do you have eggs under there? No aggression. Ta-da! She literally lays an egg on the back of the other females, kind of like, get off my back. A little nibble on the back, and then away she goes. It's just bizarre. What are females doing? Why are they laying eggs in each other's nests? But why are some females attacking each other vigorously, perhaps even killing one another, while other ones are nesting together peacefully? What's going on with this breeding system? So it's a lot more complicated. So for a number of years, with my colleague Bruce Lyon, since 2014, we ended up just going for an NSF grant, and we thought, let's try to get at the bottom of this behavior. There's a lot more to it than sort of meets the eye. So we've been doing a number of things, looking at parentage, and we've been using genetic techniques to try to determine who's doing what to whom and why. I'll tell you a little bit about that tonight. We've been doing a variety of other studies looking at the cues that they might be using. We have some evidence that wood ducks may be capable of counting, for example, counting the number of eggs, some sort of idea of numerosity, other cues that they might be using, so the cognitive aspects of this behavior. I've had other students looking at the endocrinological aspects, and I'll tell you just a little bit about whether females might be loading their eggs up with hormones, doping their eggs, so to speak. And then some video problems here. Hopefully this comes back. Wow. Let's see if we can go back here. Yeah, I haven't had that happen before, so bear with me. Apologize. And then some of our last questions deal with population dynamics. And I end the talk today trying to bring this back to a management perspective. That is a big part of my job. I'm certainly interested in behavioral biology and population biology, and also want to bring this back into some conservation implications. So I'll just touch on a couple of these things today. But when we started this project, it was designed not as a research project, but primarily as a large-scale internship project. I had all these students on campus in our wildlife major, and many of them didn't have any opportunity to get some hands-on experience. And wood ducks are great. Unlike, say, mist netting passerines, you've got to be well-trained, you've got to be careful. Wood ducks are pretty tough. Even their ducklings, they're pretty robust little buddies. So it's a great thing for students to learn how to band a bird, how to handle a bird, how to take all the measurements, egg measurements, looking for nests. And we've been doing this for 23 years. We've had over 600 students now go through the internship program, and kept it going until last year with COVID. We had to stop it, but we're hoping to get it back this year. And it's just been a wonderful teaching opportunity. And that's really why I started working on wood ducks when I came to Davis. It was just a great opportunity. They're local. The students can actually be working on them during spring quarter while they're still in classes. And they learn all sorts of wildlife techniques and measurement techniques and bird techniques, banding techniques, radio telemetry. And all they get out of it is a t-shirt. So they're doing this, and every year, the students have a competition. They get to design the t-shirt each year, and so they get a free t-shirt. At one point, we actually had 100 undergraduate interns. I had 3 graduate students managing it. It just got enormous. It's great. And so we've also had a great team of wood duck graduate students. Going back to Terry Jensen, who started all of this, working with John Anderson, and also Dan and Sarah Hurdy. I saw Dan on the line there years ago when I first arrived, and then all the way up to students that have working with me now. And I just want to point out Eli Bridge and Bruce Lyon on the right. They're my collaborators on this project, and we've been fortunate to get funding through a number of different sources. So that's sort of some of the background of sort of why we're here and what we're trying to do with this project. Oh boy. I'm going to jump in and tell you a little bit of some of what we found. So most of our work is based around Davis. So here's Davis down here on the map on the right. You can see where we work along Puda Creek. We work along Russell Ranch. We were working at Hedgerow Farms until all the water went away. We've worked at Conway Ranch and Roosevelt Ranch and up at Birdhaven Ranch as well, up in the northern part of the valley. So our first question was, who's doing what and to whom? So let's just figure out a little bit about what's going on in this system. We've used a variety of molecular genetic techniques. I won't go into the deep details. It's a little bit sort of arcane, but we started off with some pretty simple techniques, and then we started using microsatellite markers, and we had 19 different markers. And now we're using 8,500 single nucleotide polymorphisms, et cetera. All of these techniques give us a pretty precise capability to be able to estimate who's related to who, who's the parent of what offspring, and even other questions about population genetic structure. So what we can do now with molecular genetic techniques quite easily is really quite phenomenal. So we've been working with folks in animal science and the Vet Genetics Lab to develop and use these new techniques to look at parentage in these birds. We now have DNA samples from over 500 females, over 5,000 offspring. And the great thing is we now have these offspring coming back. We've got 3 or 4 generations of individuals that we know who their mothers, who their grandmothers, who their great grandmothers are now. So we have a complete pedigree, which is fairly unusual for many wild bird populations. So to get to the chase, cut to the chase, who is doing what and to whom? Well, the genetic data indicate that almost half of these nests have more than one female contributing, more than one mom. So that was a bit of a surprise. It's just like every other nest has multiple females. Almost 20%, almost a fifth of the young that are raised in these populations, in our study populations, are parasitic. So they're being reared by a female who's not their mom. And in some cases, that can be as high as 70% in a single nest. Still having some video difficulties here. I wonder maybe if people could turn off their videos, that might help a little bit. Sometimes that does slow down the transmission. Thanks. And we're finding that the number of parasitic females per nest can range anywhere from 1 to 5. So it's really quite bizarre. So it's this massive shell game going on. Widespread parasitism, multiple females laying in the same nest, lots of young being raised by mothers other than their own genetic mother. So then our next step, and this was sort of the ingenuity of Eli Bridge, one of our collaborators, is well, how do we really, that tells us a genetic story, but it really doesn't tell us who's interacting with who, why they're doing it, where they're going, what cues they're using. And so we turn to this technology, which is quite well known. It's nothing absolutely novel. It's called using pit tags or passive integrative transponders. And these are just little inert glass tags, but they have a metal wire inside that can provide a series of binary on-off digits, basically, and it gives a code. It gives a unique code. So every one of these little tags has its own unique electronic code, but it doesn't emit that. You have to have a reader, a radio frequency identification device, or an RFID device, to query these little tags when they come within range. And then it will send out the code. They use this in fisheries all the time with salmon. They'll inject these little pit tags in salmon. The salmon will go downstream, and then they'll be able to read which individuals come back from what hatcheries or whatever based on reading these pit tags. Well, we found a way, and particularly Eli found a way to adopt these techniques for our birds. The big trick was finding a way to make it cheap. So these systems up until now cost thousands of dollars. There's no way we could do it. Here's a pit tag. You can see there's a wood duck with a leg band. And here's a little glass transponder, so you can see how big it is, but smaller than a dime. What we're able to do is put these readers on the nest sites. Oh boy. Sorry, that's quite unusual. And we're able to inject the tags into the female with a little syringe. We can also do it into ducklings. We did all sorts of studies beforehand to make sure there were no effects on them. There was, so we anesthetize them, just local anesthetic and just a little bit of surgical glue. They stay. It doesn't seem to bother the birds at all. They don't even notice sometimes when we put in these tags. And then we have these readers. So here's a picture of a nest box. You can see around the entrance, it has an antenna. And that goes down to this box down here, which is contains the reader. The reader is this little green card. It's just a little green computer. It's like a little CPU, effectively, that has an SD card that will record all the data. And when a female goes on and off a box, anytime a female or duckling comes in or out, the reader will query the tag that she contains through the antenna. It'll power up through the antenna. And then it will return that unique code. And then we have a record every time the female comes on or off a box. So we now have these pit tags and over 600 females, over 5,000 ducklings. And the thing was putting these readers on every single nest box. In fisheries, when they do it, they have one big massive reader on a stream. That's thousands of dollars. Eli developed these little readers where we could put one for about $30 on every single nest site. So we have them on about 197 nest sites. And now we've gotten almost 2 million reads. Hopefully this works out. We're having some video issues, but hopefully this works out. I'm going to show you 2 reads. I'll show you a number of them, but the picture on the left, this is going to be one female. Each one of these black dots represents a nest site. And this is her pathway, if you will, as detected by the RFIDs and the pit tags over the course of the summer. This female on the right, you'll see, has a different pattern. So I'm just going to play these, I hope. So you can see that the female on the left, well, except it stopped, is bouncing all over the place. She's going to multiple nests throughout the whole season. The yellow pathways just indicate her paths between the nests. The degree of red color indicates how frequently she's at any given nest site. The female on the right was only in a single nest. I'll just let this female continue on with her. All right. Okay, so here's what that female actually did. The one on the left, you can just sort of see there's this whole, these are all the nest sites, and these are all the different, journeys that she took back and forth. It's like she was weaving a sweater for Christmas. It was just absolutely remarkable. That particular female was a 1-year-old female. So this is her pit tag code, E9BA09, it's the last 5 digits. She ended up visiting 34 boxes that summer. She made 195 different visits, individual visits, including 30 to what turned out to be her favorite box. From the genetics, we know that she laid eggs in at least 4 different nests, and they were all quite close to where she'd hatched the year before, just a few boxes down, and they were all close together. She never had a nest of her own that year in 1995. She came back the next year and did exactly the same thing, even more. Now she was on 42 boxes, still never incubated a nest, still laid eggs that we could detect genetically. So she was just traveling about like a traveling salesperson, laying eggs willy-nilly in boxes. This might seem like it's unique, but it's not. So here's another example. More recently, this is 2017, I think, at Roosevelt Ranch. So this is Roosevelt Ranch up near Woodland. This is the whole wetland area. What you're going to see is a series of dots showing up that represent different females on a nest box. And this is actually just following the entire population over the summer. Different colors represent different females. Different females are going to different boxes. Some are going to a few, some are going to a lot. I mean, it kind of looks like an LA freeway in rush hour. And then towards the end of the season, it tapers off. That's what it looks like if you just sort of sum all of these visits together. We had no idea. I mean, we sort of had an idea that females would wander about a little bit. They might check out some different boxes. We had no idea there's this complexity of nest searching and egg laying behavior. I'm going to show you a couple graphs. I don't mean to be too you know, getting too much into sort of heavy duty graphics and statistics and so forth, but hopefully this will be fairly straightforward. On the bottom, I'm showing different females. These are their pit tag numbers. And this is the number of the boxes that the female was recorded in. These little red bars just indicate the number of boxes that each one of these females was captured in. So that was a nest where we actually caught her on the nest. And that's up until now, that's the only way we could capture females. That's the only way we could ban females. This is our standard practice to get an idea of what's going on in a population and who's in that population. You see that a lot of these females weren't even caught. Most times we would catch them in a single box. Every once in a while, we catch a female in 2 boxes. That's based on just the good old fashioned way of catching a female in a box. Here it is when you now add in the data that we've got from the radio frequency identification devices and the pit tags. I mean, it's just astounding. It's a completely different pattern. There's females that are visiting 51 boxes. And there are a lot of females, for example, this group here, we never caught at all. And yet they're very active in the population, going to 20, 15 boxes. Here's sort of a classic example. This other female right here that I highlight here, she actually went to 51 different boxes. We never caught her, but she clearly was an active member of the population. So we're getting a very different story about what these females are doing, how they're traveling along boxes, how they're finding nest sites, and whether they're present or not. And it's giving us an entirely different view of the structure of the population. So had we just been doing it by the old fashioned way, by capturing them on the nest, we would have got about 60% of the females. Almost 40% of the females were never recorded on a box, on a nest box. They were only recorded using our pit tags and our RFIDs. So they were there, they were alive, they were contributing to the population, and we never would have counted them, we never would have considered them. So that's hugely important in terms of the population dynamics, but it also says there's a big segment of ghost females out there that are being active and doing things we can't imagine. It turned out this is another graph. This is what we call a histogram. These are the number of different boxes that a female was recorded on, from anywhere from 1 up to 51. And this is just, you know, the number of different females that were observed on 1 box, on 2 boxes, on 3 boxes, etc. And what you find is there's a group of females that are really just only on 1 or 2 or maybe 3 or 4 boxes. They're staying fairly site faithful. They're not wandering about. And then there's this other group of females that are just roving all over the place. They're going in 20, 30, 40 different boxes in a breeding season. And we know they're laying eggs. They're not just looking for nests. There is some nest prospecting for sure. This red figure here, part of the histogram, would be, this is what you would expect this pattern to look like if you just randomly took all of these females and just, you know, threw them in boxes, like throwing marbles into a bucket. Some would have a lot, some would have a few, but this would be the distribution you'd expect. What we're seeing is very, very different from random. We're seeing a group of what we're sort of referring to as stayers and a group that we're referring to as rovers. There seem to be 2 flavors. I mean, it's a continuum, but there seem to be 2 flavors of females in these populations. And now with our genetics, what we're finding is that many of these nesting, the host females that are on a nest that we do catch are also visiting other nests, and they're also laying eggs. About 39% of those females that have a nest of their own are getting something else on the side. They're also laying eggs in somebody else's nest. So it seems we have three different categories of females in the population. We have one group that are only laying eggs as parasites. They're never incubating a nest on their own. We never catch them. There's a second group of females that are the stay-at-home females. They only have their own nest. So this is sort of what we would think about as the sort of prototypical bird. But that's only about 23 to 30% of the females. And then there's a third group of females that not only have a nest of their own, but they're also laying some eggs parasitically. So they're doing both. So we have these three sort of flavors, and they're about equally common. The ones that nest only, and from our genetics, we don't have evidence that they're laying eggs anywhere else. That's about 37%. The ones that are doing both nesting and parasitizing are about 39%. And the parasitic only females are about 23%. So a completely different view of what they're doing. This is kind of statistical. I apologize. It's from a sort of a more, it's from a scientific meeting, but this is the number of ducklings produced over these females' lifetime. So this is a sample of females that we've been followed from when they were first-year-old females to when they've died. And it's usually 2 or 3 years. It's their sort of maximal lifespan for a lot of these females. Here's the females that only laid eggs parasitically. This horizontal bar represents what we call the median. So that's about sort of, it's like almost like the average. It's close to being the average. It's sort of the halfway point. The boxes represent sort of the major portion of the distribution from the 25 to the 75%. And then these are the ranges, and some of these are outliers. The numbers you might want to look at are the numbers at the top. This is the number of ducklings on average that a parasite-only female would produce in her lifetime, 6.4. So she's getting something out of it. It's not zero. There are some that get zero, but some that are doing pretty well. The females that only have a nest of their own are doing better. They're getting about 2.5 more ducklings over their lifetime. And the females that are doing both are doing even better, up to 14. And you can see that some of these are doing extraordinarily well. There's females that are getting 50 or 60, producing 50 or 60 ducklings over their lifetime. So there are some real super mums out there. So what it looks like is there's kind of a gradient. And so this is where we've sort of where we're at now in terms of our thinking about what wood ducks are doing. It looks like there's a group, and we're still testing some of these hypotheses, but they look like it's mostly young females. These parasitic only females seem like they're young females. They may be in poor condition. It may be a case where they can't get a nest site or food resources, and they may not be as experienced. And so what we're seeing is it seems like it's the young females that are doing this parasite only tactic. Then there's females that seem to be in good condition. These are our nesters. They can get a nest site. That's kind of the status quo. That's what we had thought of as being sort of normative behavior. But then there's these super mums that are doing both, nesting as well as laying some eggs on the side. They seem to be females that are really high quality. They're in good body condition. They seem to be older females, more experienced, and they're getting an enhancement. They're able to lay more eggs, and perhaps more eggs than they could rear themselves. Or maybe what they seem to be doing is laying some eggs parasitically, taking a break, and then starting a nest of their own. So they're almost double clutching without having to care for 2 broods. So that's kind of where we're at now. There's still some more pieces to fill out on that part of the story, but there's a twist. Waterfowl, as many of you may know, are unusual amongst birds in that it's the females that are the philopatric sex. It's the females that come back to where they were hatched to breed. In most other species of birds, it's males that come back to their natal area. In waterfowl, it's females. That raises an interesting possibility, and it may be that these females in the population are in fact closely related. So maybe we've got it wrong. I've been calling it parasitism, as have everybody since Yomtov 1980 onwards, but maybe it's not. Maybe this is just big family daycare, and it's mothers and daughters or sisters or granddaughters and grandmothers that are all interacting together in a collaborative, cooperative way rather than a competitive way. It's a twist, a complete twist. So maybe this is back to the days of, peace, love, and music, Woodstock. Maybe this is really communal living. And, we do see these females, like I showed you in that video, that are very peaceful, there are also females that are fighting to the death. So this raises sort of a deeper question about the underlying social dynamics of these birds, the social network. You know, of course, we're all deep into that now with social media and Facebook and Google and so forth. Well, birds have most animals, many animals have their own social networks. And we're only now beginning to apply the same tools, the analytical tools that have been developed in the social sciences, been applied by Facebook and Google so they can track you and your sales and your purchases. But wood ducks often hang out together, particularly when they're looking for nest sites and perhaps we think when they're laying eggs. So we can use our RFID data, these pit tags, to begin to uncover the social network. And what we're finding is wood ducks remind me sadly of my high school days. There are cliques. So this is just a small network. These are a group of females. They're connected by who they hang out with. And we record that by their RFID reads when they're visiting the same boxes together in the same time period. And we use a variety of statistical techniques to determine whether they actually are hanging out together or whether it's by chance. So here's just a small example. So this is a group of females, the ones in orange here, that are all quite tightly connected. They all sort of visit box together, they hang out together. These are the cool kids. And then there's a group of somewhat social females, but they're a little bit more outsiders. And there's probably what I was in high school, kind of, the loner, the outsider out tromping around the woods rather than hanging out at the parties. That's sort of a simple example. And it's actually interesting because as when I was preparing this talk a while back, I was looking for examples of human social networks, and I came across this one, and it's almost identical to this social network for this group of birds. This is on Russell Ranch. It's a small population. Here's a more complicated one. So this is a social network. Each one of these points is an individual female. The lines are connecting hens that are visiting nests together. So if they're tightly coupled, they're hanging out all the time. If they're a little bit more loosely coupled, they're not hanging out as frequently. They don't have as many shared visits. And then we can do this for all possible pairwise combinations, and that develops this network. And what you see is you get females with many connections, and they're part of that core social group. That's the in group. And then you find females that have few connections, and they tend to be non-social explorers. So we're seeing this real difference in terms of who females hang out with that could come into playing a role. And that's got us interested, and we're going back and we're actually just doing these analyses now. Things kind of got put on hold with COVID. We had to sort of shut down a lot of the DNA analyses in the lab. But this is that network. And for this 1 year so far, we've been able to pull apart the genetics. And what you see is that network is comprised of sub-networks of relatives. So these are sub-networks that are all connecting 2, 3, 4, 5, 6 females that may be related either as siblings, mother, daughters, or as cousins. So to the level of about the same as a cousin or a half sib would be. So those networks, these complex networks, when we start to unravel them, look like there could be subgroups of kin. And so maybe it's these groups of females that are being nice to each other, and it's groups that are not related to one another that are fighting to the death or competing with another. So it's a much more complex interaction, and it really does remind me so much of our own human societies, really sort of dealing with friends and families. So that's all I'll run out of time to sort of go into it, but we have 2 PhD students that are working on these projects, and the results are just still coming off the press. We got delayed a little bit with COVID, but it's really revealing a very different underworld of the wood duck than we had presumed. One of the other studies we worked on, this was actually a few years back, was there are some ideas that perhaps females might be loading their eggs with various kinds of hormones. And this idea has been around for a while. It's been shown for a number of species, particularly steroids like androstenedione and testosterone. So these are steroids. And the working hypothesis was that these, by loading eggs, and it's in the egg yolks themselves, females may be able to manipulate the development time and development rate of the embryos. In particular, eggs with higher levels of testosterone may develop faster. So with Nicole Dell, who is a PhD student, and working with another PhD student at Hubert Schwabel at the University of Washington, we explored this with wood ducks, and it's a tricky deal. You have to be able to extract some some egg yolk without damaging the egg. You want the offspring still to hatch. There's a way to do that. You have to use gas chromatography to be able to sort out the steroids. And I'm just going to show you one graph of what we found. And it really opens up sort of a whole series of additional questions that we're beginning to explore. The eggs that were laid by the parasitic females, so that's shown here in red, compared to the host females, the females that laid eggs in their own nests and incubated them, had higher levels of steroids, had higher levels of this one steroid, androstenedione, A4, was higher in the yolk. And this is a testosterone precursor, and it's actually less toxic. And so it may well be that this now gets metabolized by the developing embryo into testosterone, and that could facilitate its hatch rate. And we also found that ducklings that had higher levels hatched faster. So maybe the game afoot is that these parasitic females are loading their eggs, they're doping their eggs so that their kids can come out, kind of as super kids, or at least can develop more quickly. And potentially they may be duping the host female. They hatch out first, the host female leaves with them. That's awesome. And the female's own kids may be left behind. So there could be evolutionary arms races that may be going on basically inside the nest as mediated by these hormones. So that's another area that we're beginning to explore. Okay, my last 5 minutes. Let me close off with more of a conservation and a management angle. So the first part of that was just sort of some fun natural history, using some of these new tools with pit tags and RFID and detailed molecular genetics, trying to sort of peer into what is it that drives the social system, these life history strategies of these birds that we see every day and had no idea how complex their life might actually be. But I'm also interested in, well, what are the practical applications of this information, of this knowledge? And it may well have something to do with how we're managing their habitat, and particularly how we're managing nest boxes. So this is a common pattern. This is from the Great Swamp National Wildlife Refuge. Oftentimes when wood duck programs are established, they take off. So this is a graph in the light blue here is the number of eggs laid over a series of years. And it goes from 0 to almost over 15,000 eggs being laid. And that lasts for almost a decade. And likewise, the number of ducklings shown in the dark blue here, that also increases. So it looks like everything's going gangbusters. The nest box, these are nest box populations. And then you get this crash. And it doesn't completely go down, but it sort of goes down, it sort of oscillates, the number of ducklings goes down. So what's going on? Why is this population sort of overshot its limits? And what started out as, sort of really high numbers goes down by to 2/3. It goes down to sort of 1/3 of what it was. Well, starting in the late 80s and throughout the 90s, Paul Sherman and Brad Semmel said, you know what? We are killing these birds with kindness. What we're doing is exacerbating this behavior that I've been talking about, conspecific parasitism. So this is a behavior they propose that has evolved in birds that where nest sites were probably hard to find, large cavities in big, vast forests. It really paid to follow other females around or to pay attention to really check out where their nests were. That could explain some of this nest ranging behavior we're seeing. But the problem is when we've added nest boxes, we thought, well, if one box is good, then 20 is even better. And we'll put them side by side. And what Paul and Brad have said is now we've completely changed the ecological landscape. These birds, if they're following each other into these nests, there's no longer any sort of hide and seek. And you're getting just, really high densities, high visibilities of boxes. You're getting exaggerated levels of parasitism. And what happens is you get these build up to these massive numbers of eggs, these dump nests, 30, 40, 50 eggs, and that can lead to greatly reduced reproductive success. So we've gone from a situation like this with natural cavities where you would have estimates from the literature, maybe less than 1 cavity per hectare. And here's a natural cavity of a wood duck that's actually had a natural nest in it. And now through our well-meant, but perhaps misguided management techniques, we've put up really high boxes, densities of boxes that are super visible, where birds can follow each other. We've sort of caused an ecological trap. And the result is you get these massive numbers of eggs. And then what happens sometimes is many of these eggs, either the nest is abandoned or there's so many eggs, the female can't incubate them and they start to rot. Some are okay, some are bad. And our own data indicate when you get clutch sizes that get too large, hatch success really declines. So this is just a graph showing the proportion of eggs in a clutch that are hatching as a function of clutch size. And these are just averages for different nests with 5, 10, 15, 20 eggs. And hatching success is around 70% until you get to about 25 or 30 eggs, and then it just plummets. You get beyond 30, 35 eggs, it's just zero. There's not even any variation there. There's just no nests hatch out. It looks like this. So you get these really high densities, and that leads to these really large clutch sizes and nest success declines. So one of the things we did when we set up this internship, we said, well, let's actually test this idea. So that was, sort of part of the questions that we started out with back in 1998. We established a number of boxes, a number of sites, Puda Creek and Russell Ranch. We also had Hedgerow Farms, but it unfortunately went dry. So the population there was sort of extirpated. And Conway Ranch and Roosevelt Ranch, we had a low density site where we tried to hide the boxes. The densities were low. There was a couple hundred meters between every box. And then we had high density sites where we had lots of boxes per unit distance, and they were very close together. So now with our RFIDs, we can actually really dive in and ask whether the behavior of the birds differs depending on the density of boxes. So here's Conway Ranch, where we have high density of boxes. Roosevelt, we actually have high density locations and low density. So that's where we were trying to do a control within the same area. And then here's Russell Ranch, where we have a low density and all these boxes are hidden. And you can see very clearly under the high density situation, that's where you get these tons of these birds are going all over the place, mass, 50 birds, 40 birds visiting a nest box. You see lots of birds as well in the high density areas, but not so many on the low density areas at Roosevelt Ranch and Russell Ranch, where it's just all low density. You're still getting, multiple birds per nest. That's a normative part of their behavior, but it's nowhere near the same. How does that affect parasitism and reproductive success? That's what we're concerned about as managers. So here's our high density sites from 1998 up till 2015. The high density sites had a greater proportion. This is the proportion of parasitic eggs shown in red here, had a higher frequency of parasitism than the low density site. So that's exactly what Paul and Brad had predicted, that you get a lot more of this parasitic behavior when you've got these really high density sites. What about hatch success? Well, as expected, these high density sites also had lower hatch success on average. So that seemed to support their idea that when we put up these really high density sites, we get a lot of this behavior and it impacts hatch success. I'm going to come back to this, pay attention to this oscillation in this high density. But here's the manager's dilemma. When you actually look at the total number of ducklings produced from these high density sites, these same sites that I just showed you, and the low density sites, not looking at the per nest, but the total production of ducklings, you actually get more when you put a higher density of boxes. You're getting more eggs laid, fewer of them are hatching proportionally, but the total number of ducklings produced is higher in these high density sites. So that's the manager's dilemma. What do you want to do? You put out a low density situation, you're going to have less parasitism, higher hatch success. You'll have fewer nests and fewer eggs laid, so you're going to get lower overall total production. If you're just trying to produce ducks, this isn't going to be as successful, but it's going to be probably more characteristic of what a natural situation would be, or a more historical pristine situation might be. Under high density boxes, you get a lot more parasitism, hatch success goes down, but you do have more nests because you've got more boxes, you get more eggs being laid, and even though fewer of them hatch proportionally, you still get a greater total production. So that's kind of the manager's dilemma. And we were left with, boy, what's the answer? Our bigger concern, and this is where I'll finish off, is that really it's not even just what's going on in total production, it's what happens with population stability. So the idea you could get this high density, you get frequent parasitism, that leads to lower success. Maybe that drives the population down a bit. So now you've got lower density. Maybe that causes less frequent parasitism. So success goes up, that builds up to high density. So you could get these kind of cycles developing that are driven at a population level purely by this behavior. And so I just go back to this graph that I showed you with parasitism. You can see under these high density situations, you kind of get these oscillations, which is consistent with this cycling idea. And we've actually now done some formal mathematic modeling, and it gets sort of pretty deep into the stats. but we've shown actually that this behavior parasitism can cause population cycles, and if it gets too extreme, it can cause populations to crash. So that could explain some of the phenomena that other people have seen. So that's I don't have time to go into all the aspects of what we're looking at. I just kind of wanted to give you a flavor for, some of the things we're finding with the new technology. It's a lot more. Their behaviors are a lot more complex than we thought. We're still trying to get a handle on the mechanisms, differences among females, the relative importance of these social networks, and whether it's kin-based, relative-based or not, and whether moms are playing games with hormones in their kids, and then ultimately whether this is impacting the management population dynamics. So, there really are duck secrets. I mean, wood ducks have been studied, for almost a century, and yet there's still things that we're discovering. So, when we look below the surface, we can sometimes see a world we would never imagine. There's still secrets that can amaze, impress, astound, and confound us. And we don't have to look further than our own backyard. So I am still amazed. I hope you are amazed as well, or at least knowing a little bit more about these birds that are in our backyards. Yeah, if you are interested in this, I did a couple of podcasts with the Ducks Unlimited podcast, so you could go and search them online. And there's two sort of 40-minute podcasts that I did with Mike Brazier, sort of talking about this in birds, this behavior in birds in general, and then a little bit more details on this study with wood ducks. So that's it. There's just a lot of people that have been involved in this project. We're still going on. I'm only a year and a half from retirement, so I'm not sure how. We probably are not going to figure out all the secrets of the wood duck world before I retire, but hopefully I'll have some graduate students that will continue that on. So thanks very much for listening. Sorry for some of the technological glitches, and I'd be delighted to take any of your questions. There we go.
Thank you, John. That's pretty interesting. Who knew?
Yeah, exactly. Yeah.
Yeah. I was, throughout it, I was wondering about, oh God, with what's happened to the habitat in terms of preferred habitat for wood ducks. How do you figure out what the carrying capacity is for what wood ducks need when you have such a proliferation of nest boxes and that high density producing high numbers of ducklings? How many make it to adulthood? So that's for your successor, I guess. I mean, unless you can do it in a couple years.
I mean, we're getting. I mean, that's where the pit tag technology is really coming in, Ken, because there is estimates, and actually, it's really interesting with regard to the carrying capacity. So Gary Hepp has done some work back east. They're not using pit tags, but they would put little fish fingerling tags, web tags on the webs of ducklings. But that means you have to capture them again in hand. So only when the ducklings come back, recruit into the population, the female ducklings, catch her on a nest, you know that she was a duckling. And they've estimated recruitment using that technique. They estimated that recruitment was not high enough for eastern wood duck populations to be able to sustain the populations, the ones that they were studying. There had to be some sort of immigration in order to maintain the local population based on its reproductive rates. That struck me as surprising, simply because where are these birds coming from? They were studying a fairly typical population. So were they just studying sinks? I mean, where are the sources? When we apply our RFID technology, we're finding all of these ducklings coming back. We're not catching them on the nest. That's that 39%, but they're coming back and they're laying eggs. So they're actually adding to the reproductive potential of those populations. It changes the lambda, the population growth rate. And when we use our recruitment rates, the populations are sustainable. So it's very different depending on the technology, because we're underestimating recruitment using sort of the standard techniques we used to use. Right. With this tool now, we can begin to look at how those recruitment rates will vary depending on nest box density. We can look at how that changes over time in terms of population stability. So carrying capacity isn't just about reaching the maximum. It's also about how variable it is. You don't want big swings where you can, drop off the face of the cliff and crash. And so that's that's what we can get with some of these new data. We're getting far better demographic information to be able to estimate carrying capacity. There's a big question of to what extent do boxes actually contribute to that. Frank Belrose estimated back east that maybe they only contributed 10% of the recruitment. He didn't think that that was the saving grace. He argued, and I think rightly so, that what we need to do is replenish the habitat. We need those older growth forest. And I would say the same thing for California. Riparian restoration is critical, not just for wood ducks, for all sorts of other
Right.
riparian birds. Migration, I mean, it's just on and on and on. You all know the importance of the riparian habitats, and particularly where you get old enough riparian forests so you can get these large cavities. And that's going to take some time. So I think nest boxes are a stopgap measure. I would not want to advocate, hey, here's our major conservation tool. It helps, but ultimately we want to get back to replacing the habitat.
Mhm. Okay.
Sorry, long-winded answer. Yeah, I warned you before. Ask an academic a question, and you're going to get 6 answers.
Yeah. Okay. Zane, there are a few questions in the chat, and Zane, can you read some questions, please?
John, so the first one that I'm seeing is, what is the threshold percentage of the sequence difference required for individuals to be determined to be a subspecies? If you know that off the top of your head.
Not if, I mean, yeah. Oftentimes in the past, we would use sort of pretty standard sort of population genetic analyses. And I can't remember what the, there's a variety of statistics that we'd use. The FST is sort of a common statistic. I can't remember what that actually is. I think it's less than 0.1, which is just sort of an index. And I can't remember offhand what that is. Yeah, no, I'd be sort of pulling numbers out of the air. So, basically, and the way, I think there's sort of far more sophisticated techniques that people are using these days with, sort of almost whole genome kind of analyses, and there are thresholds that are being set. So I don't know the percentage sequence divergence that is being required. So there's thresholds that are being developed based, I think, on empirical data. So what do we see in terms of well-established species, subspecies, et cetera? And then based on those kinds of distributions, when we find a new population and we're asking, is it a subspecies or not, we're referring back to those thresholds. I don't do a lot of sort of phylogenetic work, so it's work with colleagues that I've done that have sort of said, hey, we actually have a genetically differentiated population, but it's not at the level of a subspecies. So sorry, that's a bit of a vague answer, but it sort of gets a bit beyond what my expertise is.
All right, and well, how about for the RFID devices? Have they been used with natural tree holes? So it's just wanting to get an idea of whether there might be any effect on the artificial boxes and their close proximity with this behavior.
Oh yeah, yeah. Oh, I'm sure. I am absolutely sure there's. I would love to be able to do that, and you could certainly do it. It wouldn't be a problem at all. It hasn't been done. We've looked for natural cavities in our populations. There's just not that many. It's really hard to find them. So yes, you could certainly use them. It hasn't been done. Ours is the first study that's ever been done using this technique for wood ducks. There has been some work done on European species, pied flycatchers. I think actually it's getting more and more common. Tree swallows, people have done some work on tree swallows. Those are all with boxes though as well. So the natural cavities would be great. That would be the next step. You'd have to have an area where there were enough natural cavities that you could really get access to them and get the RFIDs on them. But the technology would certainly be suitable. It wouldn't be a problem at all. Yeah.
All right, and the next question says, do individual females seem to follow the same behavioral style of the 3 that you listed across their lifespans, or do you see a change from some individuals? That's a great
question. Yeah, we've actually now been able to look at that. And hi, Kirk, how are you? Boy, that was from Kirk, who was an undergraduate and went on to do great things and actually worked with on Sarah and Dan Hurdy's property on turkeys, and actually did a great study on kin relationships amongst male turkeys that was just fantastic. Anyway, so hi, Kirk. Yeah. So it's a mixed message. Females are switching behaviors over time. So it's not a fixed trait. It's not like a female is parasitic for their entire life, but it gets tricky. So what about a female like that one I talked about, E9, who lived for 2 years and she was parasitic in both years? Well, what if she'd lived for 3 years or 4 years? Might she have changed? We do know that females will change, and they seem to follow this trajectory from being parasitic in their first year to nesting or doing both in their third year. And then they seem to drop back to either nesting or parasitic when they're older. So it seems to be an ontogenetic kind of trajectory. Having said that, we do see some females that seem to be following kind of consistent pathways throughout their life. So it's not hardwired, but there is predilection. So I think there's personalities, and we're looking into that, looking at the repeatability of parasitic egg laying across years. The other thing that tracks is I mentioned the stayers and the followers, the female or the rovers, stayers and the rowers. We're finding fairly high repeatability of those behaviors as well. So females that are roving in 1 year, going all over the place, tend to do the same thing the next year and the year after. So there certainly seem to be strong, or there's Sarah, strong behavioral traits behavioral, I'll call them personalities. And we're looking at, so I've had another graduate student, Mitch Hinton, who's been looking at early growth, whether it could come back to the hormones, it could come back to early lifespan, it could go back to growth rates. Those patterns seem to continue throughout these birds' entire ontogeny and in captivity as well. So it's mixed. It's not hardwired. There is flexibility, but there's also a high level of repeatability in many of these behaviors. So hopefully
Very
that
nice.
sort of answers the question.
And then two of these questions are kind of related, but someone says, are there different genetic markers for the 3 different behaviors?
Yeah.
And also, would long-read genome assembly possibly reveal something fundamentally different about the
Yeah.
individual styles?
Yes, to the latter would be really interesting to look at. I suspect so for the former, but we'll see. I mean, that's going to be for my whoever follows on after me. I mean, we now have the genetic tools to be able to do deep dives into the genome and look for, sort of not just markers, but sort of, whole segments of the genome that may be correlated with and may even be partially coding for specific behavioral variants. I mean, they're really where we've got in terms of behavioral genomics in the last decade has just been phenomenal. So I suspect so. That's getting a bit beyond my abilities, but I think this would be a great system to explore that. And we're finding that in a lot of other systems, so it wouldn't surprise me at all.
Does there seem to be a variation in lifespan amongst the 3 nesting behavior categories?
Yes, although we're just getting that information now, we're getting enough variation. It really does look like some of those sort of super females, they're living longer, they're producing more kids, they're also laying eggs parasitically and not parasitically. So it really does look like there are females that and even from a fairly early age, and we suspect it's a lot of this is correlated with early age growth rates, sort of, and what mom did. I mean, so these are maternal effects that are the transgenerational that are translating in these females. It's the silver spoon kind of thing. And so they happen to come from a big egg, they're a big duckling, they grew fast, and then they sort of are riding the Mercedes into the rest of their career. And then, so that's a simplification, but yeah, so yes, I think there are genetic markers. I think there is going to be just, variation between them. It's not going to be fixed, but there's going to be strong influences of these traits in combination with environmentalists, genetic and environmental interactions that I think that are determining these pathways. Interface that with shifts in the population size. So there's a population behavior feedback loop. Sorry, I'm going to get a little bit deep on this, but one of the interesting things, let me just play this out. Imagine a high density situation, lots of competition for nest site. That favors a certain type of phenotype, a female that might be more aggressive, like those fighting females we saw. Perhaps, I'm just sort of speculating. And so that could also facilitate maybe either, testosterone levels rise incidentally, or maybe females that have, have higher levels of androgens, load that into their eggs. Those kids are more successful in that highly competitive environment, but then the population goes down, right? It's that it follows that cycle. And now you're kind of selecting for a different type of female, maybe the females that are more collaborative and more cooperative rather than the ones that are highly aggressive. And so it could be those population dynamics led by the behavior itself that could be selecting for different types of females over time. So there's this feedback loop between the behavioral dynamics and the population dynamics that I think is really fascinating.
That's super cool. How widespread among waterfowl do you think this type of parasitism is?
Very widespread. In fact, so I think of, there's 160 species of waterfowl or so, and I think it's been recorded in well over 80 species. So waterfowl are notorious for this. They really are. They're the dump ducks. I mean, as are, it's actually interesting, as are galliforms. So, grouse, quail, etc. And Galloanceae are sort of this ancient group of birds, right? This sort of pretty early diverged group of birds that's broken off or not, or that is different from the rest of the avian clades. And maybe so I fantasize that this behavior was found in theropod dinosaurs. So you never know. That's a bit of a fantasy, but yeah, so it's very common in waterfowl.
That's cool. And kind of a follow-up question to that is the parasitism within waterfowl, is it primarily cavity nesters, or would that have to do with there being scarcer amounts of available nest cavities?
It is more common in cavity nesters, but it's not exclusive. So there are overwater nesters and ground nesters. It occurs in mallards, but the other notorious parasite in North America is the redhead. And so it will lay its eggs, and canvasbacks as well, but the redhead is quite parasitic as well, and it will lay, and it's an emergent marsh nester, an overwater nester. So it's not just the limitation of cavity nests, but most of the cavity nesting waterfowl do in fact show this behavior. So certainly it could be resource limitation, it could be the ease of finding your host. A cavity nest is just going to be a lot easier, and it doesn't go away. It's there every single year. Whereas a ground nest, you have to search for those nests because they're built anew every year. So it may be that some of the frequency is related to the ease of finding a host, not just to limited cavities.
All right. And how far are the females traveling when they visit other nests? I think this came up when you were showing that video.
Sorry, the scale was on there. I mean, that's those at Roosevelt Ranch, they're going 3 miles. I mean, they're just taking daily trips. We're so blinkered, and we think that these birds just going next door and going to a few nests, they're traveling all over the place. These are migratory birds, so I think our understanding of their sensory perceptual abilities and just even their information base upon which they're sort of making reproductive decisions is far different than I think we realize. So they're going 3-4 miles, and that's partly just a limitation of where we have our nest boxes. If we had our nest boxes over a 10-mile square area, maybe they would be going that far. It takes a few minutes to fly for a duck to fly from one end of Roosevelt Ranch to the other. So yeah, they're ranging quite widely.
I think you touched on this at the end, but could nest box placement and spacing from each other affect the parasitism?
Yeah, absolutely. Both. So there's the density, which is kind of the spacing, like how close is it to a neighboring nest? But there's also sort of the placement. So the other aspect of that is the visibility. However, we have looked at a bunch of habitat characteristics, including visibility of the nest site, amount of vegetation cover. That influences whether a nest box is used for the first time when it's put up anew. But after that, it's a social tradition. What best predicts whether a box will be used as not habitat is whether it was used last year. So yeah, I liken this to pubs on, the Davis Cat, like a pub where everybody flocks to one, and right next door is a pub serving the same beer, playing the same loud music, but nobody's going to it. So it's these social traditions, sort of a follow the leader kind of response. And I think maybe that level of information may be useful. I mean, when you think about this ancestrally, finding birds who have knowledge, using them as conspecific information sources, conspecific cueing could be really important when you've got a scarce resource that's distributed in a massive hardwood forest. So that sort of behavior could quite well have been selected for. And I think maybe now the way we're putting nest boxes up, we're sort of, that behavior is now being sort of exaggerated, sort of co-opted by our placement, not only our spacing, but also by our placement, high visibility. So it's both.
Very cool. In reference to those rover or ghost females, since they're not sticking to any nest box, where do they roost during the breeding season?
Oh, they're resting. The wood ducks are not roosting in the nest boxes either. I mean, they'll be nesting. when they're not incubating. I mean, by and large, they'll be, they're nesting on the water, on logs, etc. They're pretty flighty, as most people know. So they'll be, yeah, I mean, they'll be roosting, and they may be roosting together. I mean, we don't know. That's going to be interesting to find out, is who they hang out with when they're not on the nest, kind of thing. But yeah, no, so they're roosting. They're roosting elsewhere. They're not in the boxes themselves.
This one says, if you ever capture a female, I'm guessing they're referencing like an adult, how do you track them?
Well, because we capture them as ducklings. I mean, that's the whole, innovative aspect of this new tracking. So if we can trap or capture most of the ducklings that hatch, now there may be some in natural cavities, or there could be immigrants. But if we can capture the ducklings, we can put a pit tag in them. They're not losing their pit tags. And so even if we've never caught her, we can track her because she's got her pit tag, at least in the boxes that we're following. So I think that's one of the innovative components of this. We have females that we've only ever caught, if you will, as a duckling when they hatched out within 24 hours of hatching. And then we've never actually even seen them, but we know they're there because we've got their reads and we've got their genetics. So, certainly there are some females that come in from other areas, no question, they're immigrants. I think there's one or two natural cavities that we haven't found yet, because we always find a few, birds that seem to have been hatched out just recently, but they're not from any of our nest boxes. So it's not perfect. It's not perfect, but we're 90%, I think, which is quite unique.
That's nice. Kind of following up on that, it says, do you have evidence of females that fly longer distances and may fly into and out of your study sites, such as our edge effects?
Yeah. Hi, Sid. Yeah. Actually, Sid, surprisingly not. Because we've got birds on Puda Creek. We had, several places along Puda Creek, including Russell Ranch, where you did all your fantastic Swainson's hawk work. And then also, up at Conway Ranch. And we've also got, hunter returns from these birds, and they're hanging around together all year round. The fact that we also are finding some level of genetic distinctiveness between these populations, even between Conway Ranch, which is just up in Woodland, and Russell, which is just down by Davis, which means that probably the males are being somewhat philopatric as well, which is not really expected in order for there to be lack of gene flow. So that's really suggesting that there's not much immigration, that these really seem to be functioning like what we call a meta population, somewhat distinct little populations, which I find really surprising. We don't capture birds that have been banded elsewhere, even when we've been banding, or other organizations like Cal Waterfowl have been banding. We're not capturing a lot of other birds. Every once in a while, we'll get another banded bird. But other than that, however, we do find birds that are unbanded in our boxes, particularly at Roosevelt. So it seems like that's an area where there's another source population that we're not necessarily tracking the birds. So we don't yet have a sense of the population level structure. That's one thing we're hoping to do. There's a bunch of nest box programs that CWA has monitored for years. And if we can get genetic samples, we can begin to use genetic tags to look at, estimates of population flow, population turnover, immigration and emigration, and effective population size. So yeah, it'd be really interesting to look at the social network writ large, across the whole valley.
That's nice. Let's see. Oh, are there any other cavity nesting waterfowl in California? And if so, what are they and where?
Yeah, there are. Hooded mergansers are becoming increasingly common. Actually, they're starting to show up in our boxes, so that's probably the second most common. You get bufflehead further north. You might get some common mergansers. I don't think they're super common. So I would say it's mostly, if we don't get common goldeneye or Barrow's goldeneye. So I would say it's mostly hooded mergansers by far, and then bufflehead would be the two. Not bufflehead down in the valley, but you get them up in Northern California. So those would be the two more common cavity nesting waterfowl. Yeah.
Very cool. And then the last one that I'm seeing is, does your genetic analysis tell you anything about the role of males in the population genetic structure?
Yes, it does. Yeah, thanks, Sid. Absolutely, it does. So what we're finding is males are sneaky folks as well. I mean, so the crazy thing with this mating system is females are mating with multiple males. and males are mating with multiple females. And I'm not sure whether it's polygynandrous or whether it's just promiscuity. There's a variety of different mating systems. So we wondered whether, males might mate with multiple females and then they would both lay their eggs in the same nest, something called quasi-parasitism. We're not really finding evidence of that. What it looks like is sequential polyandry and sequential polygyny. In other words, females will mate with multiple males for different sets of eggs, but even eggs laid in the same nest may have multiple sires. And then males may mate with multiple females in different nests, perhaps being laid at different times. So with the genetics, we're finding that indeed there's just a lot of skullduggery going on, both males and females.
That's very
Thank you,
interesting.
Zane.
Yeah.
Thank you,
Thank you,
Zane.
John. I think that's it.
Yeah. Yeah.
Okay. It'd be cool to learn duck language, and those females who are incubating together, if in their small talk, they both happen to drop the same male's name. Yeah. Well,
Yeah,
yeah.
absolutely.
So it was, I mean, we've got some thank yous coming in, and I want to thank you again for a wonderful and enlightening talk. It's just something, I knew about canvasbacks, redheads, ruddy ducks, but the wood
Ruddy
duck,
ducks is another. Yeah.
it was just so cool. This how sneaky they are. Yeah, and I love.
Yeah,
the
we've known that they do this behavior for some time, but had no idea just sort of how expansive and how complex it might be. And I think we're just now starting to realize all the interesting pathways one could go. I wish I'd done this at the beginning of my career rather than at the end of my career. So So yeah, for the Kirk Emsons and the Harnawas and the Camerons, there you go. Some of my students, I saw Doris on here as well. So here you go. Here's your well, Kirk is pretty well established now, but here's I throw the gauntlet to you if you're at all interested.
Yeah, excellent. Anne, do you have anything that you'd like to close with?
I'll never get my audio back. I mean, my video. No, except that I did realize that when I talked about the December 8th monthly meeting, that it's going to be given by Emmett Iverson. I think I never said his name because I saw Steve Hampton there and forgot about Emmett. But Emmett is the one who does our monthly column on the recent sightings in the newsletter, and he does a super job. So it'll be a good presentation.
Emmett will lead the bird ID. And thank you for saying that, because I wanted to remind folks that December 8th, we have the bird ID wide workshop for the Pewter Creek Christmas count. And yes, as Ann said, Emmett Iverson will be leading that. And let's see, I don't think there's any other announcements. I want to thank everyone for joining us tonight. And thank John once again. It was a wonderful talk. I had no idea it was going to be so
bizarre,
eye-opening. Eye-opening, I tell you. Yeah. And the graphics and the numbers everything was able to understand, put some perspective into what you're saying. And thank you for spending so much time to make this presentation that we could all understand and enjoy, and we'll look at a wood duck differently when we see one next. All right, everyone.
Thanks. It's great to see all the names and see some faces. Hi, Sid and Alice, and everybody. It's just that's heartwarming. I look forward to seeing you all in person as I get closer to retirement. I will become more involved in the Audubon group. That's
great. That'd be great.
So,
yeah, we can meet at that pub with not that many people at it, because there'll be more chairs.
yeah, we'll be the loners. We'll be the outliers with those few social connections. Yeah.
All right.
Fantastic. Thanks
everybody.
everybody. Take care.
Thanks,
Have a good night. Stay
everyone.
safe.
Thank you.
Happy holidays.